Good evening. Today I will brief about the mass spectrometer in our lab. So this is the mass spectrometer. This is ORBITRAP from Thermo Scientific. The model is ORBITRAP EXPLORIS 240. 240 signifies the resolving power or resolution of this instrument is 2,40,000. Basically, this instrument is regarded as HRMS, high resolution mass spectrometer, with ORBITRAP technology. This is the patented technology of Thermo Fisher Scientific. So high resolution means in this mass spectrometer the resolution can go up to 2,40,000, but the variants of ORBITRAP has different resolution or resolving power. So high resolution in proteomics study is very important because we look for low abundant proteins in our sample apart from high abundant proteins. So to detect and quantify those low abundant protein, high resolution is needed because the high abundant peaks masks these low abundant protein peaks. The best example of this is plasma, where the albumin and immunoglobulin has high abundant peaks, but several other proteins or crucial proteins also there which can be identified and which are of definitely in low abundance, so they masks the low abundant proteins. So to separate them out, high resolution is needed, but too much high resolution is also not recommendable because ions, the mass spectrometer detect ions. So if resolution is very very high, then the loss of ions will be there, and which in turns give not a good result. So the resolution has to be optimized with respect to the maximum resolution. So here we optimize the resolution in 1,20,000. So not too much high or not very low. If low resolution is very low, then there will another problem. So we have to optimize it, and here we optimize it into 1,20,000. So this is the ion source. This is nano source. So now in this mass spectrometer, in generally there are three parts: ionizer, analyzer and detector. So this is the ionizer. The ions are generated from here, goes inside the mass spectrometer, separated by quadrupole, and then analyzed and then detected. So the ORBITRAP is a technology of Thermo. So the ORBITRAP technology or ORBITRAP instruments has a specialty that in conventional mass spectrometer, ionizer, analyzer and detector are separately arranged, but in ORBITRAP the analyzer itself acts as a detector also. That means here no separate detector. The ORBITRAP itself trap the ions and detect the signals and give the results. So there are lots of videos and PDFs in the internet regarding ORBITRAP. So I am not going so much details of that part. I have only briefing how the mass spectrometer looks like and how it works. If you want to know the structure of ORBITRAP, it looked like this. a barrel shaped, centrally thickened, and two sides are narrow. This is the shape of orbit trap which is inside this instrument. And orbit trap is also analyzer, and the mass spectrometer makes depends upon this analyzer, because different types of analyzer are there, and based on analyzer mass spectrometer are making. That means TOF analyzer is there, this 3D, 2D, ion trap, and lastly the orbit trap. Orbit trap is the advanced of ion trap. So what I am saying, this is the nano source. So in mass spectrometer, ionization source are of two types, MALDI and ESI. MALDI means matrix assisted laser desorption ionization, and ESI means electrospray ionization. So this is a kind of ESI or electrospray ionization. Here two source is supplied. One is called HESI and one is called NSI. HESI means heated electrospray ionization, and NSI means nanospray ionization. So HESI source we are using during the time of instrument calibration, and nano source we are using during the sample acquisition. So now sample acquisition is going on, so we are attached this nano spray source. This is the manual injector which we are using during the calibration. So this is the ORBITRAP EXPLORIS. There is a camera in the nano source which show the spray quality, and it is the temperature of the column, which is 50 degree, and this tubing is called a nano tubing. This goes into the LC and attached to the column compartment in the 10 port valve, which I have shown in the last video. So we are now we put our samples. These samples are first go into this sample loop, and from here it will go to the trap column, which I have shown in the last video in the column chamber, and from there it will enter into the analytical column, and through this tubing it will enter into the instrument. So the tip of the column looks like this. Here the camera is there. So this is the tip of the column. This is maintained in high temperature around 300 degrees centigrade, and from the tip of the column the sample is being sprayed like an aerosol spray, and this is the ITC, ion transfer capillary. So the ions can enter into this capillary, and this is the junctional point of ionizer and analyzer. From here it will go inside the instrument, and there the separation is being there by the quadrupole, bent flatter pole, then C-trap, then it goes to the orbit trap for detection. So this is how the orbit trap instrument works on, and in our facility this is the ORBITRAP EXPLORIS 240. So it is combined with this LC system, which I have given a brief in my previous video. So those who are not aware of the nano system, please watch that video. And today I am giving a small brief of ORBITRAP MS, and if you want to know more, then you can have the ORBITRAP paper. There are very common research papers are also there where ORBITRAP instruments were used. So this is for today. Thank you, and be ready for the next video. Thank you.